Hi, my name's Anna Neumann,
And I'm Kate Roy.
and the title of our lab is How the Color of Light Affects the Rate of Elodia's Photosynthesis.
A little bit of background revolving this experiment is about the main environment of aquatic plants and how the environment is a crucial aspect to having adequate photosynthesis output. The main plant we are focusing on in this experiment is Elodia, since they have a great structure, which we mentioned later on, and to test how aquatic environments affects the rate of photosynthesis, we'll be using different colors of light to see how the plant reacts to those different environments, and we'll be able to measure how much oxygen they can produce from the photosynthesis, since the different colors can obviously trap, different amounts of light and energy and possibly cause a different result based on the plant and the different color.
So, a little bit more background in the introduction portion of our lab was about photosynthesis and the plant Elodia. So, photosynthesis is a necessary process which converts light energy into potential energy and enables the growth of plants. Elodia is a very common aquatic plant, but it actually lacks stomata, which is a structure that deals with gas exchanges in most plants. But in Elodia, they actually have something called parenchyma instead. So parenchyma, as shown in the picture at the bottom of the introduction section, that's what parenchyma kind of looks like. And through these little holes, gases are able to diffuse into the water and then enter through the parenchyma right through there of the Elodia, and from there they can travel through the air, through the plant's air channels. So the main question in this lab is whether different colored water would affect photosynthesis of Elodia. Materials and methods. The materials we used for this lab were two 5 milliliter graduated pipettes, rubber tubing, a test tube, a beaker, light bulb, test tube clamp stand, water, and colored dyes. And we used blue, green, red, and yellow. In the method portion of this lab, for each of the colored dyes, we used the same experimental setup. In order to calculate the photosynthetic rate of Elodia, a sprig of the plant was placed into a 5 milliliter pipette inside of a test tube with water. And so to just create a picture of this, we had a test tube filled with water, and then inside the test tube, we had the 5 milliliter pipette inside that test tube, and then inside of the pipette was the sprig of Elodia. And then next to the test tube was a beaker filled with water that contained a type of dye. So for each lab, we would either have, we used red or blue or green or yellow for each. And then we did each color. We did 3 trials for each color in order to calculate the photosynthetic rate of Elodia. But on the other side of the beaker was a lamp or a light bulb that would be turned on for 5 minutes for each measurement of oxygen produced. All 4 of the colored water solutions would receive 3 measurements for oxygen production for a total of 15 minutes of photosynthesis per color.
Going on into the results section of the experiment, you can clearly see that there's a table up in the right-hand corner that is labeled with each color, and you can see how each trial is measured in the 5-minute intervals, and along with those measurements is also the wavelength for that color as well, so you can kind of correlate those together. But the main aspect we are taking away from this experiment is the average values of oxygen from each color, and you can see for red it's about 0.0613 milliliters, and then yellow is about 0.186. Green was about 0.0123 milliliters, and blue is 102 milliliters. And to put put that into a better visual, we made a bar graph, but instead of having the oxygen output in milliliters, we put them into liters just because those bars would have been very, very tiny to see to the eye. But other than that, all of that is the same. And you can clearly see that blue light had the largest oxygen output, but also had the lower wavelength, which is very interesting for this lab to observe that difference. And we'll be diving into that deeper into the discussion section. But you can see that red and blue light have the most common oxygen output to be more compared to yellow and green. Going into the discussion section, this experiment offered a lot of valuable information towards aquatic plants and the environments in which they thrive in. So basically, a plant that has more red light or blue light in its environment, it's going to produce a lot more oxygen output into its surroundings and have an overall better thriving of photosynthesis for itself and throughout its life cycle. The most interesting aspect of this lab was the wavelength. Since red has a wavelength of about 665 nanometers, and and that's a larger wavelength which can actually break through water more easily and flow through and be able to absorb into the plant. And the blue light had a wavelength of 470 nanometers, which is very short, but a main thing to know about blue light is that it has higher energy potential. So as that is coming through, as light is shining through blue, it can translate that higher energy into the plant and be able to, the plant be able to use that resource to produce more oxygen. So it was a very interesting lab to go through and witness how that was. And that was the biggest takeaway that we got from the lab. But overall, the red and blue color produced the highest oxygen output compared to yellow and green, even though they were like right in the middle section, but they just didn't have large enough wavelengths or energy.
And that concludes our presentation.
Yep.